forests mostly today. If you have questions or something, just let me know at any point or afterwards. And since I'll talk mostly about our work in urban forests, I just wanted to tell people a couple of other things that we do in my lab, so that if you are interested in those other things, you could chat with me later or email me. So today, we'll talk about urbanization and climate change on trees and tree pests. We also work on the effects of urbanization and climate change on other ecosystem services. We work with dung beetles, we work with pollinators, and trees and other things, ants somewhat. So if you're interested in that, and we've done a lot of work recently on native pollinators, so native bees and butterflies and things like that. So if any of this interests you, feel free to contact me. We can talk more about that. So I like to read about old scientific things. This is my friend Vince. Vince is also from the U.S. This actually isn't Vince. This is a gentleman named Joseph Putnam who lived over 100 years ago, and he was an interesting natural scientist. He died when he was just 26. But before he died, he did some work on scale insects. So how many folks are entomologists? Anybody entomologists? A couple? Okay, so I'll spend more, I just like to know what people know about insects. These are, if you're not familiar with scale insects, this is actually an insect, and the insect lives underneath of a waxy cover, and I'll show you more pictures of those. But why I was interested in Joseph Putnam is that he discovered over 100 years ago in Iowa that these scale insects in the city that he lived in were very common on the trees in the city. But so they were very common on city trees. When he walked in the woods and took walks and picnics and things that scientists used to have time to do, then he could never find these scale insects in the woods. And so it's one of the first examples that I could find where the pests on urban trees are more common and more severe than pests in forests. And so this is the scale that looks like the one that you found. It's different. This is Bulbinaria, a different species. Yeah. Okay, so this is the town that he lived in, Davenport, Iowa. You can see this is 1890. It's not a big city like we have today, Lisbon or what have you, but you can see how the trees are in the same poor condition as we have. Okay, since then, lots of people and some folks here have studied why pests are more common in urban plants than they are in natural forests. And so lots of examples in this recent review paper. And we care about this for a lot of reasons. One, we plant trees and other plants to look beautiful. We want to make the cities and our yards and landscapes nice. We also want to reduce the amount of spray and other application of insecticides or fungicides and things like that, because of the risks that they involve to people and other animals. And trees in particular provide lots of ecosystem services to the people that live in cities. So we've worked for several years now, why are pests more abundant on urban plants? This is the basic question that we've been working on. And there's lots of ideas about this. One idea is that when you go from natural habitats to urban habitats, that plant stress increases, and that this plant stress from living in urban areas can make the plants more nutritious for pests and can also make the plants less defended, so their defenses go down. Some folks have studied the natural enemies, the predators and parasitoids of pests, and one hypothesis is that, as you go along this gradient, the number of predators and parasitoids is lower. The other things that we've been working on more is that when you move on this gradient, temperature increases. So cities are hotter than the surrounding rural areas. And this is largely due to impervious surfaces. So impervious surfaces like roads and sidewalks and rooftops that capture solar radiation and release that heat back into the environment. So one of the hypotheses that we've studied in my lab for several years is that heat, so higher urban temperatures, are one of the driving factors of pest abundance on urban trees. And so whether it was Davenport, Iowa 100 years ago, this is Times Square in New York today, we've done some work up there that high urban temperatures are responsible in part for pest abundance. Okay, just real quick, the people who have done most of this work, and when I say we, I really didn't do any of it, of course. A little bit, I make the money, and then these people do it. So this is my former student, Adam Dale. You'll hear about his work. He was a PhD student in my lab. He's at the University of Florida now. My PhD student, Emily, she finished her PhD last year. She's a postdoc at Harvard now. And my research associate, Elsa Youngstead, did a lot of this work. And this is my collaborator, Rob Dunn, who's a professor in biology. Okay. So I'll talk about three main objectives, three little stories about our research. The first thing we did when we got this idea is we just wanted to go out and determine, does urban warming affect pest abundance? If it didn't, I wouldn't have had much to do the last several years, so you can assume that that probably happened. So how do pests and warming affect tree health? We're primarily, ultimately we're interested in the trees and whether they're growing and performing the services they're supposed to. Then the last thing I'll talk about, we're very interested in whether we can use the effects of urban warming on plants and animals to predict the effects of global warming and climate change. So we'll talk about that at the end. Okay, another old picture. This gentleman was the department head in my department 100 years ago, and he was the last person on Earth to study this insect. This is called the gloomy scale. Okay, it's another scale insect. Underneath of this shell is a tiny insect, no legs, no eyes, no antenna. It just sucks juices out of the plant. So he was the last person to work on this in 1912. So I figured that there's probably still some more to learn. This pest is very common in the United States. And you can see this is a red maple, Acer rubrum. And all the bumps that are on that branch are the scale insects. And this is the way most red maples look in the southern United States, in cities with that many insects. Okay, so we know from the old work something about the life history. It has one generation per year, this insect. I guess this is how they used to deal with this in 1912. And this gentleman spraying this tree while this guy stands below him pumping the thing. And so that's the old way. We'll see if we can come up with a better way. Okay, so with this blooming scale, the way that we approach this is Raleigh, North Carolina. This is where I live. Okay, and this is a thermal map taken with the Landsat satellites of Raleigh, North Carolina. You can get these for anywhere, I believe anywhere in the world. They're free. But what it shows is the relative temperatures of different parts of the city. And just as an example, this big blue spot is a state forest, and right next to that is the airport, which is, of course, just impervious, hundreds of hectares of impervious surface. And those two are about 10 degrees C different in relative temperature. And you can see how quickly it transitions from one state to another. And so we work in the downtown area, and what we were able to do, the city of Raleigh has a tree inventory. So every tree that the city owns is referenced with a GIS location. And so we can map these green dots, our trees. We can map that on top of the thermal map. And then we were able to find trees that might be in a cool location that are very close to trees in hot locations. So we looked at hundreds of trees all over the city, about 100 trees. And just to get to the point, as temperature increases about 2.5 degrees C, scale insect abundance increases about 200 times. This is a log scale here. Okay, so over that temperature gradient of 2.5 degrees, you get many, many more scales. And that means trees look like this. This is called gloomy scale because gloomy is like sad, scary, and these trees look gray and crusty from all the scales when they should look nice and like that. So the next thing that we did, I'm going the wrong way here. Okay, so we found this relationship with temperature. And of course, the temperature is driven by impervious surface cover, roads and sidewalks and buildings. And so we found that temperature can affect arthropod. I mean, everybody, this is just our basic understanding of ectotherms. Temperature can affect growth and reproduction, winter survival. And impervious surface, though, can also cause drought for trees because their roots are underneath of roads and pavement, and the water cannot penetrate those surfaces. So often urban trees are drought stressed also. Okay, here's an example of one of our trees, and you can see the situation there. And so the next thing that we wanted to do was try and separate the effects of temperature that we found on our trees from the effects of drought that could also be caused by impervious surfaces. And so my student Adam, he went all over the city and he found pairs of trees. And so 2 trees at each site. He had about 30 sites. And one of the trees we would put these watering bags on. So these are sort of plastic bags that you fill with water. It trickles, the water leaks out slowly to water the tree. And the other tree would have no water, or just regular rainfall. And this way, we could have hot trees and cool trees that were either watered and not watered. Okay? Okay, here's Adam filling up one of these water bags. So every tree that we water got 150 liters of water per tree, 2 times per week, every week from May through August in 2014 and 2015. Okay, we had a big, this is really a lot of work. We've got this big thing you have to fill up from these fire hydrants. So we, in all of these experiments, we record the temperature of the tree canopy. So we try and get an idea of the temperature where the scales actually live. And so these are data loggers called eye buttons. They're in these little plastic cups, and we've secured those to the tree branches. Okay, and that's how we record the tree temperature. Okay, water potential is a measure of drought stress within the tree, so it's a measure of the xylem pressure, the pressure of the water, or the pressure of the water column as it moves through the tree. And so there's Adam doing this, and this helps us determine if our watering treatment actually reduced the drought stress of the trees. And if anybody has questions about any of the methods or anything, let me know. We can talk more about that. It tends to get boring to talk about methods and stuff. So anyways, here's the water potential. So again, this is xylem pressure within the tree. More negative is more stress. Okay, so more drought stress. The dotted line is the unwatered tree, and the solid line is the watered tree. So over our temperature gradient of 2.5 degrees, our unwatered trees were more drought stressed than our watered trees, but drought stress on all the trees increased with temperature. So 2 parallel effects. So the next thing that Adam did, Adam was an amazing student. He worked very hard. He always had great ideas. And he texted me this picture one morning, on a Sunday morning. And I was in my yard playing with my kids, and I got a text from Adam. So I assumed either he wrecked the truck, or the lab was on fire, or something like that. But he had spent all night figuring out how to dissect scale insects and count their embryos. And so he took this with his phone through the scope, and each of these is an embryo of those gloomy scale insects. Now the scale insects are 1 millimeter, and so you can imagine how small these are. Anyways, this is what Adam did. And so this way he could look at the fecundity of the insects as it's affected by temperature and drought stress. And so here's the results. Again, if you want to see, this is the gloomy scale insect with its cover removed. Okay, and so again, this is about 1 millimeter. It's just a glob. It's just a glob. There's no features, no behavior. It's just a little drinking machine. So here's our again, temperature on the x-axis, number of embryos per female on the y-axis. And so the first thing is that these lines are parallel. Temperature increases fecundity regardless of water. But the fecundity increases more on unwatered trees than on watered trees. Well, it doesn't increase more, it's higher. Okay, so the slopes are equal, the intercepts are different. Okay, so now we've found that there's an effect both of temperature and of drought on fecundity of our scale insects. So it looks like we're making progress on one thing. rural trees, it's hotter, it's drier, and the scales produce, 30% more eggs. Okay, tree functions. Of course, we're interested again ultimately in the trees. If the trees are covered in scales, but they function and they grow just fine, great, have at it. So we measured water potential, photosynthesis to model conductance. There's Adam again doing all these things. And this is a picture Adam used in his PhD presentation when he graduated. I hadn't seen these pictures before, but here's my expensive Lycor machine that he's balanced on top of the truck, and he used a cinder block, a brick to bend down the branch and hold the thing. Anyways, he made it work. He always made it work, so this is how he did this. The essence, I won't show you the figures, but heat and drought reduce the model conductance. So the water that moves through the stomata, it reduces photosynthesis rate, and that water increases these back to more normal functions. Okay, so tree condition. We use tree condition in the U.S. Arborists, people who manage urban trees, have a rating scale for trees, and maybe this is similar here. And they can very quickly look at trees and rate them as either poor condition, fair condition, good condition, or excellent condition. And this just gives them an idea of the general health of the tree. So for example, this tree is in excellent condition. It has a dense canopy, a nice shape, lots of leaves. Here's a red maple in poor condition. Some dead branches, a thin canopy that is transparent, and this sort of thing. So the city of Raleigh has rated all of the trees as either poor, fair, good, or excellent. And so from this, we now, for all the trees in Raleigh, have their relative temperature from the satellite map. We know their location. We know the amount of impervious surface around every tree from impervious surface, other satellite imagery, and we know the tree condition. Okay, so Adam worked on this paper. These green dots are all the red maples in Raleigh. Okay, so every red maple is here, and again, we know the relative temperature and the amount of impervious surface for those trees. And this is a logistic regression figure. It's kind of wacky. But this is log scale abundance on the x-axis and the probability that a tree is in one of these four conditions on the y-axis. So the gist of this is that as you move from zero scale abundance to maximum scale abundance, this blue, which is the poor condition trees, increases. So the probability that a tree is in poor condition increases with scale abundance, which we know increases with temperature. And then the excellent little sliver, you only have excellent trees at low scale abundance. Okay, again, with all the trees in Raleigh, just another representation of this. Tree condition, either poor or excellent. If you take the hottest and the coolest quartile of trees, you have more hot trees that are in poor condition and more cool trees that are in excellent condition. Okay, so to the extent that this condition rating Also represents growth and photosynthesis and carbon sequestration. We know that the hotter, more infested trees perform worse and probably have a lower survival. Okay, so this is my student Emily. She was also fantastic, and she worked with a pest of oak trees, another scale insect, Parthenolecanium quercifex. This is what it looks like on oak trees. All of these large brown warts are the egg cases of that insect. Okay, here's the different stages. There's Emily with her most common piece of equipment, and the nymphs, the adult females, eggs, and there's Emily counting and counting and counting. So just a summary here. The first thing Emily did was similar to what Adam did. She just went out and found the hottest and coolest trees that she could, counted the scale insects, and here with the cold trees and the hot trees, she has 8 to 12 times more scales on her hot trees. Okay, so we see the same phenomenon with this scale, which is in a different family as we saw in the gloomy scale. Emily also spent a lot of time measuring tree functions. So this is photosynthesis rate in July and then over the course of a whole year. And same thing, about 2.5 degrees of temperature difference, and photosynthesis rate declines. So the trees are not acquiring as much carbon as it gets hot. So the main thing I wanted to mention with Emily's work, she also used the city database of trees. Again, all those black dots are all the willow oaks. This is Quercus bellows, and she used the temperature of those trees, and she knows she measured the relationship between. She measured the relationship between her experimental trees. She had about 50 trees that she measured for 3 years on how temperature affected growth, diameter, basal area, essentially. And so she knew the relationship between temperature and basal area growth from her experimental trees. She applied that formula to all the trees in Raleigh, for which we knew the temperature and the basal area, to predict how much carbon sequestration would occur and how much carbon sequestration we were losing because of high urban temperatures. Okay, is that clear? It was barely clear to me when she explained it. So anyways, we have these allometric equations that describe the relationship between basal area growth and carbon sequestration. Emily used magic also, so this was amazing to me. But so the essence of this is that the predicted carbon sequestration, well, of her trees, the coolest trees in the city, is here. The hottest trees, and you lose about 12% of carbon sequestration over that temperature range. Okay, and so I think in the city of Raleigh, this was like 26. I've got the actual figure. A lot of carbon. Now, it's a lot for the city from one tree species. Compared to a rural forest, is it a lot? Not really. But it is, to the extent that these urban trees should be providing this service. And then, of course, the photosynthesis rate also relates to their other services like air filtration, cooling through transpiration, and that sort of thing. Okay. colors. So finally then, we're very interested in working on whether what we see in the city translates or can be used to predict the effects of global climate change. We're not allowed to say that in the US anymore. Sporadic weather events. Okay, so my research associate, Elsa Youngstead, did this work. And I've been lucky to have some very good people. Elsa did this work. She disappeared for a few weeks. A couple years ago, I didn't know where she went. I didn't really notice that she was gone. But she came back, and luckily she came back, and she'd been to herbaria all over the eastern US, visiting their red maple collections. Okay, so this is still red maple, Acer rubrum, and it turns out that you can find these herbarium sheets, and here's one, and if you look on there, you can still find the scale covers. So the covers of the scale insects are inert, waxy material. It's not chitin, it's wax and other things. Anyways, even after the insect dies, that sticks on the tree. And so she could find herbarium specimens over the last 100 years or so and count the scales on there. So this blew me away. Of course, Elsa, she came back, she told me what she was doing. It was just great. Okay, so she found herbarium specimens from all over. This is the southern US. There's Raleigh, North Carolina, at the top of the slide, and throughout this area. Now, the colors on this are the average temperature just due to latitudinal and altitudinal changes. Okay, so it's warmer in the south and in the east. When you go that direction, it becomes mountains and cooler in the west. So what Elsa did is she plotted this y-axis is gloomy scales per 2.5 centimeters, which is an inch. You know, that's why we do it that way. Every paper from the US has 2.5 centimeters. That's their thing. So gloomy scales per 2.5 centimeters on city trees. So these are our trees from the city, and gloomy scales on her herbarium specimens per 2.5 centimeters. Okay, the black dots are the city trees, the blue dots are the herbarium specimens. So what you find, Elsa, this is a complicated analysis, but essentially the shape of the response to temperature. These temperatures for the herbarium specimens were taken from the PRISM database that's out of the University of Oregon, and they have a whole database of historical temperatures that you can find for all over. So she used the temperature of the location in the year that the specimen was collected for this axis. Anyways, the shape of the response is the same. Of course, the magnitude is very different. Okay. Another way that Elsa looked at this was to plot, here's her specimen range by decade. This line is the running decadal average, the running 10-year average temperature of her specimens. And what you can see is that there is, of course, natural variation in climate over time. You have hot decades in the pink, you have cool decades in the blue, a hot decade in the 60s, a cool decade these. And so the diamonds on this map correspond to this axis, which is the proportion of herbarium specimens that had any scales. So the proportion that were infested, as opposed to having 0 scales. So in the hot 30s and 40s, about 30% of the herbarium sheets she looked at had scales. In the cooler time, it came down to around 20%, back up to around 40%, and back down to around 20% by decadal changes. So of course now, things are even hotter, from here on. So Elsa was curious what's happened in the last 20, 30 years. So Elsa traveled, she disappeared again. She took a camping trip around North Carolina, and she went back to any of the forests that she could find that were still forests. A lot of them in North Carolina and other states are gone. But any she could find that were still forests, she went back to those sites and resampled the trees. And so she would randomly take a branch, she brought it back, she taped it to an herbarium sheet the way it would have been. She dried it, she pressed it, the whole thing. Elsa's very thorough. And so each of these lines connects an herbarium specimen with Elsa's new collected specimen. So out of her 20 sites that she revisited, then 16 had more scales now than they did before. 2 sites had the same, and 2 sites had less. So that's how that was. So from these different analyses, we think that there is some similarity between what the temperature effects that we see in cities and what could occur from climate change. The extent and the magnitude of these changes we're not sure of, but cities may be valuable to help predict which pests will be a problem and where. Okay, so implications. Of course, if you're managing urban trees, you may want to do more scouting, more inspection of hotter trees. The pests become active earlier in the year, so this could be important. And all of this could mean that there's higher costs associated with managing those hotter trees. For people in forests, this is Raleigh, North Carolina. Right now we have about 50% forest cover, tree cover, not forest cover, in the city. This is Queens, New York. It's a much older city. They have about 9%. Okay, and so it's very hard. City canopy cover tends to only decline. It starts out okay and it gets worse and worse as you go. So, we're trying to figure out how to help preserve some of this. Of course, trees also host a variety of wildlife and biodiversity that's important, and they protect people from sun and temperature related health problems, as well as asthma and other things. Okay, so forests are just generally in trouble. Not to end on a sad note, I don't have a positive message. This is the western U.S., the mountain pine beetle, where climate change has increased its range into naive forests, and so it has caused millions of hectares of